And now, Dr. Burzynski, as you know, stressing a lot of our guests are coming from foreign countries, and certainly was originally from Poland, but I do want to stress that he's from Houston. So he lives down there, and in fact, I'd seen him many years ago knowing Jim Weiner in Pittsburgh, right, Dr. Burzynski? That's where I last saw you. Let's give him a nice warm welcome this morning. Dr. Burzynski, talk about antineoplastins. Good to see you.
Thank you.
Good morning, ladies and gentlemen. I'm sorry to do it for you on beautiful Sunday morning, but what I'm going to tell you today is just simple pure heresy. It's a heresy not only for well-established Orthodox doctors, but I'm afraid also for these brave doctors, heretics, who came for this meeting today. Well, basically what I dare to propose is that the immune system in our body is not everything. There is another body defense system completely different than immune system. This is the system which I discovered and named biochemical defense system. There are certain basic differences between immune system and biochemical defense system. As you can see, the immune system consists of certain chemical components which are proteins, which are large molecules. Such proteins consist of antibodies, interleukins, interferons, complement system, as well as lymphocyte receptors, just to mention the most important chemicals. The main mechanism of defense is destruction of invading agent or defective cell. There is another system of defense in our body which is completely different. It's a biochemical defense system. The main components of this system are peptides and amino acid derivatives. Well, there is certain similarity between proteins and peptides, because both proteins and peptides are built from amino acids. Amino acids are building blocks of both of these types of molecules. However, peptides are much smaller molecules. They contain less than 50 amino acids per molecule, whereas proteins have more than 50 amino acids. They are simpler molecules, and among these peptides and amino acid derivatives which are discovered, also various components, such as antineoplaston A, antineoplaston L, antineoplaston H, and antineoplaston O. Antineoplaston A is the largest group of these peptides, and it can be subdivided on various fractions, like from A1 to A5. Then A2 can be subdivided in A10. Some of the metabolites of A10 also have prominent anti-cancer activity, and I named them correspondingly antineoplaston AS21 and AS25. The mechanism of defense in this system is completely different. It's reprogramming of defective cells. It's no longer killing of the cells, but changing the program inside defective cell, which means that the cell will begin to function normally. And in case of cancer, for instance, if all of the cancerous cells will be reprogrammed and function normally, then ultimately we won't have cancer anymore. Why then some of the people are getting cancer in spite of having in the body effective mechanism which can change the cancer cells into normal cells? Because a number of people have deficiency of the components of the system which are antineoplastons. Some people are deficient in antineoplastons, and such people are vulnerable to develop cancer. If we supply some antineoplaston to these people as medicines, then theoretically we should get rid of cancer, and we should be able also to prevent cancer. The research on biochemical defense system, which is basically the system of antineoplastons, began about 23 years ago. At that time, I was a student in a medical school. Really, since I was a teenager, I had a great interest in chemistry. It started initially in my mother's kitchen when I was doing various experiments with mostly disastrous effects. But later, when I entered medical school, I continued my interest in chemistry. And beginning from the first grade in the medical school, I began research in biochemistry, which is obviously a very sophisticated chemistry. It's the chemistry of our body. I was interested primarily in amino acids, because amino acids are building blocks of proteins, and everybody knows that proteins are extremely important, probably the most important molecules for the living system. I was studying amino acids in blood of healthy individuals and also patients suffering from various diseases. I was mostly interested at that time if we can use the differences in amino acids in blood for diagnostic purposes, just to try to find out, for instance, early who developed heart attack, or for instance apply the differences in amino acid levels in blood for such problems like kidney failure. I was not really interested in cancer at that time, but coincidentally I also was studying blood taken from patients. What was interesting that among these amino acids in blood there were certain substances which for about 20 years were described all over the world as unknown substances. There were 3 such substances which were similar to amino acids. They were reacting in the same way, but at the same time they were not amino acids. And all over the world the research was done in this area to try to find out what they are, but somehow without much success. There were some types of chemical UFOs which some people were seeing, some people were not seeing, and they didn't know what they were. Well, I was curious what they were, and I decided to identify what they are. And it occurred that these unidentified compounds, these unidentified chemicals, these were peptides, peptides which nobody described before. Well, my curiosity was also raised by the fact that I found that these peptides were deficient in the blood of patients who were suffering from advanced cancer of the prostate. So I thought perhaps there is a certain link between these peptides and cancer. Well, this work was done initially in Poland. I was isolating peptides from blood. I found that, frankly speaking, we are not dealing with 3 non-identified peptides, but by the time I came to US in 1970, I was able to fractionate these peptides on 39 fractions, which were never described before. Well, then I came to US in 1970. I got a job at Baylor College of Medicine, and I was surprised because soon after I began working there, I got funding from the National Cancer Institute, and together with researchers from MD Anderson Tumor Institute, we began studying these peptides in blood. We are trying to find out what they can really do in cancer. Well, we got a variety of problems because obviously it wasn't the easiest to get a lot of blood for this research. The peptides which we are studying, they existed in blood in very small quantities. We need more and more bloods. Basically, I was getting blood from my friends, but then the friends began avoiding me. So finally I decided, well, we have to try something else. And well, I studied some other tissues in the body, and we found that, well, these peptides are present in a variety of places in the body, but obviously how to get supply of the fresh brain to isolate these peptides, or liver or whatever, it was virtually impossible. So then I found that these peptides are eliminated through the body and go into the urine. So then obviously this was the most economical decision, to try to isolate these peptides from urine. But since then I got into a lot of trouble because everybody started telling, well, this is the man who is handling urine, okay? So, and when National Cancer Institute is talking about me, they still refer to the urine, even 20 years later. So anyway, I found that these peptides can be isolated from urine, and by using, of course, sophisticated technology in the United States, such as free-flow electrophoresis, the equipment similar to what was used on Space Shuttle to isolate peptides from urine, we were able to further fractionate these peptides. So ultimately we got 119 peptide fractions isolated from urine, never described before. So it looks like we are into a lot of trouble, because then obviously it was impossible to determine the structure of all of these peptides, and the advice of my good friend at Baylor at that time, Professor Unger, was such, well, we better find out which of these peptides are the most important. And that's what we did. I was just keeping isolating these peptides, giving them to the guys from MD Anderson. They were testing them in various tissue cultures of cancerous cells and normal cells. And out of these 119 fractions, they found some which had selective activity against cancer cells. They were inhibiting the growth of cancerous cells, but at the same time, they did not have any harmful effect on normal cells. And these were the antineoplastons. These were the peptides which we are looking for. So basically, soon after we began the research, we found that there are two groups of these antineoplastons which are shown on the slide. One group was named Antineoplaston A, and this group had peptides which produced activity against a variety of different cancer cell lines. For instance, Antineoplaston A had activity in tissue culture against breast cancer, against lymphoma, against leukemia, against bone cancer. But in addition to that, there were some other antineoplastons which had narrow spectrum of the activity, which were reacting only on certain significant specific cancer cell line. And that's why we had, for instance, Antineoplaston L, which had activity against leukemia, but not against any other cells. Then Antineoplaston O, activity against osteosarcoma, which is a type of bone cancer, but not against fibrosarcoma, which is another type of cancer, malignancy of connective tissue. Well, from practical reason, we decided to stick to Antineoplaston A, because we knew that Antineoplaston A have activity against a variety of cancers. So why don't we work on this first? It was impossible to work on all of these together. Well, we are trying to be as scientific as possible. Obviously, I was working at Baylor College at that time. I was a faculty member. I needed to do everything according to what orthodox medicine was telling us to do. Well, we tested these antineoplastons in various tissue cultures, both normal cells and malignant cells. Then we did some animal testing. We found that they are not toxic. We can use them in the animals without really much problem. And ultimately, there was a decision to enter certain human clinical trials. Well, this was already in 1977. I spent 7 years at Baylor. Paradoxically, when I was about ready to begin human trials, the money dried up. The grant was approved by National Cancer Institute, but they told me, well, perhaps you can get some money 2 years from now or a year from now. Well, so I couldn't stay at Baylor without money, because obviously I needed to maintain a number of doctors working with me to buy supply chemicals. Well, then I decided simply go west like a young man, and simply I quit Baylor and I moved west of Baylor to Stafford, which is a suburb of Houston, and established my own laboratory. It was a crazy idea at the time. If I would have to do it today, I would never do it, because at that time, when I was at Baylor, I was living a comfortable life, like a petit bourgeois, more or less, just going toward retirement. So then my only savings during that time was $5,000, because I was spending money also. So with this $5,000, I decided to open my own laboratory. Well, somehow the people who worked with me at Baylor, the other doctors, they joined me. We opened the lab in the garage. Finally, we are lucky because we got some additional money, and the lab grew, and finally it became Burzynski Research Institute. Well, now, obviously, we have 3 facilities in Houston and Houston area. We have a large pharmaceutical plant, which was inspected and approved by FDA. As far as good manufacturing practices are concerned. So we made a good progress since then, and we are employing close to 100 people at present, a number of doctors, engineers, and the other technicians. So we are lucky, but of course crazy, because I don't know how we did it. It was virtually impossible. But anyway, somehow somebody was watching us from upstairs, I think. So anyway, in 1977, I decided to go on my own, open my own lab. And one of the first things I did, I was trying to do what is called phase 1 clinical trials. When you try to approve the new medicine, you have to go through various phases. You have to start from phase 1. What do you do in phase 1? You have a number of people, usually around 20 patients, and you try to find out what is the toxic effect on the medicine and how high you can go with the dosages. What is the highest tolerated dose? You really don't look much what kind of effect you get for cancer, because you really don't know what are the best dosages. So anyway, we had around 20 patients in phase 1 trials with Antineoplaston A. The trials were done in one of the hospitals in Houston, 12 Oaks Hospital. And after we finished this trial, we found that the patient, first of all, tolerated the treatment extremely well. We really didn't have side effects, so to speak. And in spite of the fact that these patients were terribly advanced, they had advanced breast cancer, lung cancer, colon cancer, cancer of the bladder, a number of them responded positively to the treatment. A number of these patients obtained partial and complete remission, and some of them, 13 years later, are well and alive and free from cancer and, of course, not taking medicine anymore. So we proved the point that antineoplastons can help people who have cancer, and what was also very important, we don't have really side effects when we are using antineoplastons. Well, after that, we decided on further subfractionation of antineoplastons. We decided to divide antineoplaston A on various fractions. We applied newly available technique at that time, which was high-performance preparative liquid chromatography. And we found soon that antineoplaston A can be separated in various fractions, like antineoplaston A1, A2, A3, A4, and A5. Each of these fractions consisted of few peptides. Then antineoplaston A2 has shown the most promising activity, and that's why we decided to isolate one of the active ingredients of antineoplaston A2. I named this ingredient antineoplaston A10. We defined the chemical structure of this compound, and then I reproduced this synthetically. Well, so that's what it is. This is the structure of antineoplaston A10, which is a simple molecule. It consists of a cyclic glutamine residue, which is one of the common amino acids, which is bound to the peptide bond with phenylacetyl residue. In 1980, I was able to reproduce antineoplaston A10 synthetically. The people who participated in determination of the structure was the group from Medical College of Georgia, headed by Dr. Hendry. Well, once we isolated the first active ingredient and established the structure, we wanted to know what happened to this particular chemical when it's given to the patient. We found that during the metabolism in the body, the other derivatives are formed, which also have interesting activity. Well, one of them, of these metabolites of antineoplaston A10, was named antineoplaston AS25, and it was also reproduced synthetically. And another one, this was antineoplaston AS21. Both of these have some interesting activity. You reproduce them synthetically. And back in 1980, we had a situation when we got 5 antineoplastons isolated from urine, from A1 to A5, and 3 antineoplastons obtained synthetically. Well, again, as I mentioned, we wanted to do everything what the government, was Food and Drug Administration, required. So obviously, we went through extensive animal testing. These were both acute animal toxicity, when you hit the animals with high dosages and try to find out if you can kill them, basically. You are trying to determine the dose of LT50 when 50% of animals will die. Well, virtually with antineoplaston A10, we couldn't kill these animals. It looks like probably the best way to do it, instead of injecting this to the animal, was to take a pound of medicine and hit the animal. So toxicity was virtually none. So, then we are trying also to do chronic toxicity study, when you have certain group of animals and you inject heavy dosage of medicine every day, or give heavy dosage of medicine orally every day for about a year. And for instance, after, say, 1 month, 3 months, 6 months, we pick up some of these animals, we sacrifice them, and we found if there's any bad effect on various organs, we really couldn't see any. So then there was some difficulty to try to produce the anti-cancer effect which we could see in the test tube, because in the test tube we could easily see the inhibition of the growth of cancer cells. We couldn't reproduce initially such effect in animals which have cancer, but these were the animals which have animal cancer. Well, we looked to the literature and we found that there was certain study done by London University, and this was the study on some of the metabolites of antineoplaston A10, which were known before. Antineoplaston A10 was not known, but some of the product of the breakdown of antineoplaston A10 in the body were known. And according to the study which was done in London, they found that in various animals there are different amino acids which are taking part in the molecule similar to antineoplaston A10. Well, some of these amino acids were completely different than what exists in human. In human, glutamine seems to be the main amino acid. In various species of the animals, there were different amino acids. For instance, in mice, this was glycine, which is much simpler amino acid. In rats, it was glutamic acid. In dogs, it was glutamic acid. In birds, it was ornithine, which is still another amino acid. Well, so it was obvious that what we are dealing with are the substances which are specific for certain species of the animals, well, they were specific also for human. And the only type of the animals which have similar compounds as human were a group of monkeys, old world monkeys. But of course, I couldn't get monkeys for this trial. It was too difficult to do it. So then what we decided to do is to do the study on human cancers transplanted into the animals. Well, the most important studies in this area were done by one of the medical schools in Japan, Kurume University Medical School in Japan, and they proved beyond any doubt that antineoplastin A10, synthetic antineoplastin A10, has profound inhibitory activity on human breast cancer transplanted to the mice. In other words, the tumors were decreasing and fading away when the animals were receiving antineoplastin A10. Well, the US government got so interested in these results that they sent 2 inspectors all the way from Washington to Japanese medical school, and they spent over there at least a week trying to see what Japanese are doing. And finally, when Japanese proved them that antineoplastin A10 has the activity in human breast cancer transplanted to mice, they gave us the approval of the IND for the treatment of breast cancer. So anyway, but this was further story. What I would like to do is to keep some chronology. So after we had animal testing to show the activity, then we do some other testing, like for instance mutagenicity testing to find out if we don't have any mutations when antineoplastons are used. Such tests were done in London because we had good relationship with Imperial College of Science in London as well as Hatfield Polytechnic, and they proved that really we don't have any mutations. As a matter of fact, the number of the mutations in the specimen where antineoplastons were used was smaller than in the control group. So it looks like we can probably protect from having mutations. Then there was another study to find out if antineoplastons can produce certain teratogenic effect. That means if they can produce certain changes in fetuses of pregnant animals. Again, no bad effects at all. So then, of course, a very important issue was to find out how do they work. What is the mechanism of action? Well, I look upon cancer as the disease of information processing. Why we have cancer? Because some of the cells which originate in our body will develop according to the wrong program. Obviously, everybody has million cells which are formed in our body because we constantly have to replace the cells in our body. And these cells which develop every day, they are exposed to thousands of various factors which can damage the program which is inside the cell. This is the program in the DNA, the blueprint which is telling the cell how the cell should develop for a very primitive cell into adult, fully functionable cell. Well, this program, which is a really chemical program in DNA, is under constant barrage of thousands of chemical carcinogens, various physical factors like radiation, for instance, UV light. Well, it can be interfered with viruses. For instance, certain viruses can put their own program into human DNA. And also human stress. The stress which we see every day can also produce certain changes in the program. Well, taking under consideration that we have millions of cells which are developing every day, and we are living, frankly speaking, in the sea of carcinogens, then obviously everybody has certain number of cancer cells in our body. The cells which began to develop according to wrong program because of damage to our normal program, but all of these factors. So it's obvious that everybody would develop cancer sooner or later if we wouldn't have certain protective system in our body. And this is biochemical protective system. This is the system of antineoplastons. Antineoplastons can correct the program inside the cell and can push the cell toward normal development. Well, every cell has somewhat limited lifespan, and every cell is programmed to die. Especially earlier this year, there were some extremely important papers published in Science magazine which proved that virtually all of our cells in the body are programmed to age and die. For instance, fibroblasts, after going through, for instance, 40 cell division, will enter the stage of irreversible aging from which there is no return. Then they will stay in the stage of aging for a certain period of time and they will die out. Then the new cells will be formed to replace them. Well, what we are trying to do is we're trying to start with uncontrolled cell proliferation, which is represented by cancer cell, which means the cells which are multiplying without practically the end, are forming tumors and finally killing the body. From this stage, we are going, under influence of antineoplastons, into controlled cell proliferation, which means the cell will still divide, but they will specialize, they will undergo differentiation. Differentiation in medical terminology is specialization of the cells. Ultimately, after so many cell division, they will enter the stage of irreversible senescence, of aging, and ultimately they will die. If the cell which has programmed how to make cancer will die, the program will die along with the cell. Such studies were done by many places. For instance, we did some studies in this area in our institute, but perhaps the most important study was done by the Department of Pathology of the Department of Defense in Bethesda. They proved by using Antineoplaston AS21 in the tissue culture, that means that when we have these cells in the test tube, after approximately 2 to 3 days, the cancer cells will change into normal cells. They will behave completely like normal cells, and then they stay normal until they die, providing that we don't remove the medicine too soon. If, for instance, after a few days we remove the medicine from culture medium, they will go back to cancerous way of life and will multiply like crazy. So this means that when we are treating a patient who has cancer, who has a number of cells which are multiplying like cancerous cells, we have to maintain certain consistent concentration of the medicine, of antineoplastons, in their body, in their blood, because if we slow down too soon, then we have to start from the scratch, because the cell will begin to multiply again and literally go toward cancerous way of life. Again, this is more or less just a basic diagram how this treatment works, just trying to push the cell to behave, trying to push the cells to become normal cells, and then ultimately, when they stay normal and all of the cells will go through this route, we won't have cancer anymore. But how it works on the molecular level? Well, we found that there are 3 distinct mechanisms of action of antineoplastons. I don't want to be too technical, but I would like to give you some idea how this may work. Well, for instance, antineoplaston AS21 and AS25 works through inhibition of incorporation of glutamine into cancerous cells. But such inhibition is specific for cancer cells. What is very important for cancer cells is excess of glutamine during certain phase of the cell cycle. Well, cells have certain life cycle which is subdivided on various phases. We have phase G1, we have phase S, which is the phase where we have phase G2, finally phase M, where we have cell division or mitosis. For instance, in most of the cells, such cycle lasts approximately from 20 to 24 hours. This phase here is the most important, because here the decision is made in the cell at this moment, which is approximately after 7 hours, starting from 0 point, if the cell will enter nucleic acid synthesis, DNA synthesis. Once it will enter DNA synthesis, everything else will go automatically. The cell will divide, and if we are dealing with cancerous cells, instead of 1 cell at the beginning, we have 2 cells, say 20 hours later. Well, what we found is that if we decrease the amount of glutamine by giving antineoplaston AS21 or AS25 to the patient, and in the same way we substitute glutamine by the peptide, which will compete with the glutamine, then we push the cancer cell to behave in normal way. Then the cancer cell will not go any further. It will stop here, and it will behave as normal cell. There are various details, how it works, which reactions are taking place, but I don't want to bother you with this. I can discuss this later with whoever will be interested. Antineoplaston A10 has a different mechanism of action. Antineoplaston A10 reacts directly with DNA inside the cell. And the way it reacts, it's known in scientific terminology, intercalation. Well, this is the diagram which shows various bases of DNA. For instance, you have adenine, thymine, guanine, cytosine. Well, what antineoplaston A10 is doing is being inserted in between these two bases. Okay? And what is peculiar about it is that antineoplaston A10 we react only with certain sequence of DNA. Obviously, DNA has many sequences, but A10 will react only with certain sequence. And what is important about this sequence is that this sequence reacts with most of known carcinogens, like benzopyrene, which can cause lung cancer, aflatoxin, which can cause liver cancer. So A10, by going inside these bases, block access for certain known carcinogens. And there are various other chemotherapeutic agents which will work through intercalation with DNA. For instance, adriamycin, the mechanism of action of adriamycin is such that it will intercalate with DNA, but it's not specific. It will intercalate, it will react with any sequence of DNA, and that's why it's so toxic. A10 is not toxic because it reacts only with some sequences. Well, how it looks on molecular basis, I would like to explain through following slides. Well, this is the model of the molecule of antineoplaston A10. And again, this is the model of the fragment of the DNA. What happens when A10 enters DNA? It's been inserted into certain sequence of DNA, between certain bases of DNA. Well, again, it's a broad area. A number of studies have been done in this area, especially by Medical College of Georgia. They use both chemical studies, like influence of A10 on denaturation of nucleic acids, on melting point of DNA, and also they did extensive computer modeling. But that's basically how this seems to work. Well, there are some other antineoplastons which have different mechanisms of action. Antineoplaston A2, A3, and A5 work through inhibition of the process of methylation of DNA and nucleic and RNA in the cells. And through specific inhibition of methylation in cancerous cells, we are able to induce differentiation or specialization in these cells. Again, this is a detailed subject, and I don't want to bore you with extreme details, but I'd be happy to discuss this with some of you after my lecture. Well, so I think it's about enough about the biochemistry and mechanism of action. Now, I would like to tell you a little more about what happened when we give antineoplastons to humans. Well, back in 1980, we had quite a complex situation because, as I mentioned to you, we had 8 different antineoplastons. And each of these antineoplastons was available both in injectable and oral form. Well, so what was necessary to do next is to do phase 1 clinical trials, to do whatever we did initially with antineoplaston A. For a number of years, since 1980, we are involved in 20 different phase 1 clinical trials. Each of these trials contained from approximately 20 to 50 patients. And in each of these trials, we are trying to determine for each of these medicines what is the highest dosage we can accomplish, what are the side effects, and ultimately at the end we are trying to see what is the anti-cancer effect. Well, what we proved is that these antineoplastons did not have any toxic effect in patients. The patient really tolerated the treatment extremely well. So the side effects was not really our problem. We also found that antineoplastons have very interesting anti-cancer activity, especially in the tumors which usually do not respond to chemotherapy, radiation, and immunotherapy. Such tumors like, for instance, malignant brain tumors, advanced cancer of the prostate, certain forms of lung cancer, bladder cancer, even cancer of the pancreas. Well, out of these 20 different ways to treat cancer, we selected 8 for the next stage, which is phase 2 clinical trials. What is done in phase 2 clinical trials, we simply take a group of patients, usually from 15 to 35 patients, who are diagnosed with the same type of cancer. So that means each group will have people diagnosed with the same type of cancer. And these people will undergo treatment the same way, with the same preparation of antineoplastons, administered the same way. That means, for instance, if you like to do clinical study, phase 2 clinical study with antineoplaston A10, well, we need to do separate study, for instance, for breast cancer treated with capsules, and separate study for breast cancer treated with injections. And then separate study, for instance, for brain tumors treated with capsules, and separate study with injection. Well, we are dealing with something like about 150 different types of cancer, and it looks that in each type of cancer, ultimately, you may need to do a separate study to find out this time what is the anti-cancer activity of the substance, of antineoplastons. Of course, these studies were done on a relatively small group of patients, but the introduction to phase 3 studies, when a few hundreds of patients are involved in each category and treated in various places all over the country and all over the world. So anyway, we began 2 phase 2 clinical trials at the beginning of 1988, and we completed them already. In the first trial which we did, we concentrated on highly malignant brain tumors, which are called astrocytoma. Back in 1988, we had a group of patients diagnosed with highly advanced astrocytoma. Most of these patients had stage 4 astrocytoma. We grade astrocytomas as well as many other tumors from stage 1 to stage 4. Stage 1 is the least advanced, stage 4 is the most advanced. Well, we had basically most of our patients in stage 4. People who already underwent surgery, radiation, some of them chemotherapy, immunotherapy, and in spite of all of this, the tumor was growing like crazy. So we had tough cases to start with. Well, we treated these patients by giving them antineoplaston AS21 and A10, which are synthetic antineoplastons, in form of intravenous infusions. Well, it sounds difficult, but it was very easy. What we did, simply, we connected the IV drip through a special pump to the tubing, which was inserted in one of the veins in the patient's body. This was a catheter which was inserted for the entire length of treatment. And we connected the IV drip going to the pump before the patient was going to sleep. So the patient was receiving the IV drip, for instance, for 7 hours. When he woke up in the morning, the IV drip was disconnected and he could go back and enjoy life. Well, most of these patients were treated outpatient because the treatment was extremely easy. We did not experience any side effects, so we are simply teaching the patient, the members of the family, how to hook up the IV drip through the pump before going to sleep, how to disconnect it in the morning, and then the rest of the day the patient could enjoy life. Frankly speaking, most of the patients improved rapidly, and some of them, after, for instance, 6 weeks of treatment, were going back to work part-time. The children were going back to school. We gave maximum 6 weeks to find out how the patient was responding to the treatment. If we did not see objective response in 6 weeks, we assume that the treatment is not working. And how do we determine the response? Again, we use classical methods to measure the response, what is done by any medical school. That means we based our response on the size of the tumor. If the tumor disappeared completely and the patient did not have any sign of cancer, we call this complete remission. If the tumor decreased more than 50%, we call this partial remission. If there was less than 50% decrease of the tumor size, we call this objective stabilization. When the tumor was growing, well, this was progressive disease. Well, I'm trying to explain this in the lay terms without going into details, but I think this will be easy to understand this way. So basically, how to find out if the patient is responding objectively with brain tumor? You have to do the scan, and most of these patients had magnetic resonance scan or CAT scan before the treatment and then after 6 weeks of treatment, and then at certain times interval to find out what's going on with the tumor. And most of these patients responded very well to the treatment. Practically 80% of patients involved in the study had objective response to the treatment. And after 2 years, a number of these patients are already off the treatment, without the tumors, and enjoying life, back to work, back to school. Some patients, of course, did not respond to the treatment, and we switched them to another program of Antineoplastons, and some of those who did not respond to this program responded to another, and already obtained a partial or complete remission. Well, some patients were not disciplined enough. They took treatment only for a short period of time, then they were bored, they discontinued the treatment, and then the disease progressed. But those who took the treatment as we told them got very good results. Again, I would like to show you some of these results. Unfortunately, I did not bring the pictures of the actual patients, but I would like to show you what we see on the scans. Well, what you have here is the MRI scan of the brain. These are the fragments of MRI scan which are showing the tumor. This patient was an unfortunate 36 years old woman who had astrocytoma stage 4 of brain stem. The tumor is visible on pre-treatment scan here as the white spot. Such location of the tumor did not allow the patient to be treated surgically. She received radiation therapy, but unfortunately a few months later the tumor began to grow. Even in 6 weeks time interval before she came to our treatment, the tumor grew substantially. Well, we started her on treatment more than 2 years ago, and after approximately 2 months of treatment, we have seen some decrease of the tumor size. Then, after 6 months of treatment, the tumor was gone. And what was also very important, we could see perfect reconstruction of the brain tissue in the place where tumor existed. We don't have any holes in the brain, except for the hole this patient had from initial biopsy. So unfortunately, they took the piece of the tumor to diagnose it, and this will persist. But there was no destruction of the brain beside that. The patient returned to normal situation. The symptoms disappeared. Now she is off the treatment for approximately a year, enjoying normal life, back to work, and does not have any recurrence of tumor. Well, the studies which you see were done both in Houston by Objective Department of Radiology as well as University of California in San Francisco. Well, again, I would like to show you another example, which again, these are the CT scans of 32 years old woman who was diagnosed with astrocytoma stage 3. Her tumor was located in the place which can be helped surgically, so that initially she had resection of the tumor, she was followed with radiation therapy and chemotherapy. Unfortunately, the tumor grew back. Then she came to us approximately 2 years ago. This is the initial scan. This is the scan after approximately 2 months of treatment. And this is the scan after approximately 8 months of treatment. The tumor disappeared. What we see is certain remnant, certain scar tissue, because the patient was treated before with surgery. So she is going to have certain deficit of brain tissue because it was removed surgically. Now this patient is over a year off intravenous treatment, without symptoms, without the tumor, and living normal life. Of course, I have a bunch of these slides, and I could show you many more, but I don't want to bore you too much. So this is just to show how we measured the response and how the patient responded. Of course, the patient looked greater and greater physically. They came to us very sick. Some of them couldn't walk. Then obviously they came back to life, and they look like normal people today. Well, we found that this way of treatment, when the IV drip was administered for, say, 7 hours during the night, did not which have the highest malignancy in brain. And these were basically the tumors which are called glioblastoma. Glioblastoma is such a bad tumor of brain that patients usually don't live longer than 6 to 9 months after the diagnosis. Well, a year ago, we did not have really good success in glioblastoma, but at the beginning of this year, we used a different way of treatment. What we decided to do is, instead of hooking up the IV drip when the patient was going to sleep, we put the medicine into the pump, the miniature pump, which was attached to the patient garment, to the belt, and this pump was delivering the medicine 24 hours a day. That means that the patient could walk around with this pump, and the pump was loaded, for instance, every 2 or 3 days. We taught the family how to do it, and the treatment was very easy because the patient could really go back to work, back to school soon after that. We found that when we give the treatment continuously, the results are much better. And then we apply this treatment for the treatment of glioblastoma. what was remarkable that most of the patients who came to us at the beginning of this year were children for whom there was nothing could be done. They were just really sentenced to death. And they responded extremely well. Well, this is a magnetic resonance scan of one of these 10-year-old patients. This is the boy who initially was diagnosed with brainstem glioblastoma, which is seen here on MRI scan. They couldn't operate because of location of the tumor, so that they radiated the boy. The tumor decreased initially, then grew back. This is when he came to us. Then after 4 weeks of treatment, the tumor was gone. And also what is remarkable, we see perfect brain structure. We see every groove, every detail which is supposed to exist. Of course, the boy is back to school and living normal life. Well, we don't have yet many patients in this category because we began at the beginning of the year, but it looks like approximately 60% of these patients whom we had in the program so far responded very well, responded much faster to the treatment than the first group of patients. Well, malignant brain tumors are a very special category because obviously there is very little what can be done for these people. But this is not such a common problem like another cancer, which is cancer of the prostate. For instance, in the United States, there are over 10 million people who have various forms of cancer of the prostate. Most of these people don't know they have cancer because it takes time to develop this. And unfortunately, there is not good treatment available for that. The mainstay for the treatment for advanced cancer of the prostate is simply castration. Either this is surgical castration or medical castration by the use of medicines which are suppressing hormone action. In spite of that, the people who have advanced disease will have recurrence of the disease after a while. With the best treatment available, the average is that they will die after 34 months from the using hormonal treatment. I'm talking, of course, about advanced cases. Well, so there's practically no acceptable treatment for this disease. So what we did in the next trial, we involved patients who have advanced cancer of the prostate, those patients who did not respond to anything. They were really sentenced to death. The trial, again, began 2 years ago. The treatment was very easy this time. We are giving the treatment orally. They were simply taking capsules of antineoplaston AS21. We found that 85% of these patients responded to the treatment. How did we measure the response? We used the common scientific methods available. For instance, you can determine if the patient who has prostate cancer is responding by measuring certain tumor markers. One of these tumor markers is called PSA. And this is what happened in the patient who is treated. Usually, this tumor marker, which can be determined by blood tests, is coming down to normal level rapidly. If we did not see the decrease of this marker within 6 weeks, we assume that the treatment is not working. Well, we obviously have seen very good improvement in patient condition. The pain was decreasing, the weight was improving, the patient was coming back to life. After 3 months, we could see improvement on bone scans. This is the bone scan which shows the location of the tumors, which are black spots, before the treatment. This is what we could see after 3 months of treatment, marked reduction of these spots, and after 11 months of treatment, virtually the spots were gone. What we see in the spine, this is just the normal picture. Well, so this could be very important to treat this type of cancer because of no acceptable method available. So far I stressed 2 types of cancer. Of course, the project is quite big. We have also very exciting results in the treatment of advanced cancer of the pancreas. At this moment, it looks like approximately 60% of patients treated for this cancer have objective responses, and a number of them have durable responses. Like we have some patients who are now approaching 3 years since we treated them and are in remission. We have exciting results in the treatment of lymphomas like non-Hodgkin lymphoma, certain forms of breast cancer, bladder cancer, and also colon cancer. Well, again, in certain areas we do not have any good results, like for instance in the treatment of childhood leukemia or testicular cancer, and we simply do not accept such patients for the treatment. I would like to stress that antineoplastons have also application in the other areas, and some of the important areas is early diagnosis of cancer and prevention. Also, they can be applied to the treatment of benign tumors, like benign brain tumors, breast tumors, which is fibrocystic disease, and also genital warts. Genital warts are benign tumors or nodules, which is a big problem because there are approximately 14 million of patients in the United States who are suffering from this affliction. And again, the treatment is very easy, consists of administration of antineoplastons orally. We see improvement in certain skin disorders like psoriasis, chronic ulcers, and we see rapid healing of wounds. We also see improvement in AIDS and certain neurological disorders like Parkinson's disease. Well, as far as AIDS is concerned, we began the treatment of AIDS patients only recently, and it began with the patients who have both cancer and AIDS at the same time. Well, we found that most of these patients responded positively to the treatment, but we still have a relatively small number of patients. We began at the beginning of this year, and we have only one patient who became HIV negative at this time, but most of these patients had marked improvement in their cells. The T4 cells were increasing after the initial 4 weeks of treatment. How the treatment works in AIDS? Well, it's known that AIDS is caused by HIV virus, which is incorporating the DNA, the program into the DNA of the cell. Such cell, obviously, ultimately will have division and will yield to some offspring cells, which also will have a program for AIDS. Well, the virus will multiply and will infect some other cells. And we have certain type of chain reaction. What we do in these patients, we are supplying them with antineoplaston AS21. Most of them are treated orally in the form of capsules. Well, then, the antineoplaston AS21 will block the program which is in the cell DNA. This is the AIDS program. The cell will not divide anymore, will undergo specialization, will function normally, which is, of course, benefit for the body. The virus will not be able to multiply in the cell which is not dividing. And when the cell will die, the virus will also die. And hopefully, this will be the main benefit for the patient. Well, again, I would like briefly mention positive results which we observe in small number of patients who have cancer and at the same time they had Parkinson's disease. Again, Parkinson's disease depends on malfunction of astrocytes. Astrocytes are the cells in the brain which are producing growth factors which are necessary to maintain neurons. The most important part of the brain is substantia nigra, which is giving impulses to another part of the brain's striatum and ultimately to cortex. What happens in patients who have Parkinson's disease? They have a failure of the program. The astrocytes, the same cells which can produce malignant brain tumors, astrocytoma, do not produce enough growth factors, and the cell will die. And then we have a breakdown of the impulses here. Well, antineoplaston A5 will correct this program and force astrocytes to produce normal amount of growth factors, which again will maintain neurons. Well, there's another antineoplaston, antineoplaston A10, which will also benefit patients who have this program. But mostly, the patients who have symptoms of Parkinson's disease produced by the use of drugs. Certain patients who are using designer drugs, they will get symptoms of Parkinson's disease because of presence of certain toxic chemical. It's called MPTP, which is converted by the enzyme monoamino oxidase B into active ingredient MPP. And again, such conversion will take place in astrocytes. Well, MPP plus will damage neurons, will stop oxidation in mitochondria in neurons. And then, of course, we have a breakdown in this pathway. What antineoplaston A10 will do? Will inhibit the enzyme, and then, of course, we don't have conversion of MPTP to MPP, and we do not have toxic effect on neurons. This study was done by Medical College of Georgia and proved that this is the mechanism of action of antineoplaston A10 on Parkinson's disease. Of course, we don't have yet many patients in this category. I don't want to have excessive enthusiasm about it, but certain preliminary results are quite interesting. Well, in spite of the fact that we spent a lot of time in this area, I still feel like we barely scratched the surface. It's a tremendous amount of work to be done. We are enjoying very good cooperation with prominent researchers from the United States, from 2 colleges, United States, Georgia Medical College, and also Department of Pathology of the Department of Defense. We have very good cooperation with Japanese medical schools, also with some medical schools in France, Netherlands, Italy, Germany, and recently in Poland and also in the Soviet Union. We really hope that working together, we can solve various problems in contemporary medicine. But there's a tremendous amount of work to be done. Well, at the end, I would like to thank you very much for the attention to this program, and I would like to thank Laverne and Steve Ross for an excellent idea of putting together this foundation and organizing Congress like that. Thank you very much.
Thank you. Doctor, we'd like to give you a plaque.
Thank you very much.
Thank you very much. You're on your way back to Houston, right?
That's right.
Okay,
Love
good.
to book.
Okay, thank you. Dr. Burzynski, thank you. Thank you very much.